So, reproductive physiology. This is basically a summarized version of the Oxford Handbook of Endocrinology and Diabetes. Reproductive endocrinology, first of all, we will discuss the anatomy. The normal adult testicular volume is 15 to 30 ml, and the ovarian volume is 15 to 5 to 10 ml. LH and FSH address two gonadal cell types with feedback from the sex steroids and inhibin. Three important cells of the gonads are interstitial cells, cell supporting gametogenesis, and the germ cells. So among the interstitial cells, in male, these are the Leydig cells, and in the females, they are the thecal cells, which are found in between the seminiferous tubules and the follicles, respectively. Both of these interstitial cells produce testosterone under the LH drive. Now, the second one is cells supporting the gametogenesis. So in male, these are the Sertoli cells, and in females, these are the granulosa cells. These cells basically secrete various hormones, including inhibin and Müllerian inhibitory factor, that is AMH, under the FSH drive. So the interstitial cells are under the LH drive, and the cell supporting cells are basically under the FSH drive. So the Hormone inhibits FSH secretion from the pituitary gland, that is the inhibin inhibits the FSH secretion from the pituitary gland, and the AMH is responsible for supporting the female sex organ development during the sexual differentiation in utero. In adult female, AMH is produced in proportion to the germ cell number and is therefore a marker of ovarian aging. Germ cells. Male continue to make new germ cells throughout the adult life in the basal membrane of the tubules. Female cease to make new germ cells after the birth and therefore bond with the eggs that they will never make. Function units. In male, these are the seminiferous tubules, make up 90% of the testicular volume. Spermatogenesis occur in the presence of the high intratesticular concentration of testosterone, made up of germ cells and Sertoli cells through which spermatogonia mature to be released into the lumen of the tubule. In female, these are the graphene follicle. Primordial follicles are recruited in batches, mature over 2 months with selection of the dominant follicle with single central oocyte surrounded by the granulosa salve, which convert theca-derived testosterone into estradiol for ovulation. Follicles which do not proceed to ovulation becomes atretic. So causes of low sex hormone binding globulin. All the causes are related to increased insulin resistance in body, like economically, growth hormone use, Cushing syndrome, anabolic steroids, hypothyroidism, and obesity. Causes of increased sex hormone binding globulin are cirrhosis, estrogen, tamoxifen, phenytoin, and hyperthyroidism. So regulation of the gonadal function. Basically, there are a few structures which help in the regulation of the gonadal function, including the hypothalamus, pituitary, gonads, and then the regulation of the gametogenesis. In the hypothalamus, gonadotropin-releasing hormone is secreted by the hypothalamus in the pulsatile manner, and the stimuli are from the cerebral cortex and the limbic system through the various neurotransmitters, including the leptin, kisspeptin, endorphins, catecholamine, and dopamine. GnRH release occurs during sleep, in early puberty, and then throughout the day in adulthood. It stimulates the secretion of luteinizing hormone and follicle stimulating hormone by the pituitary gland. The pattern of GnRH secretion is crucial for normal gonadotropin secretion. Faster pulse frequencies are essential for the LH secretion, whereas slower frequencies favor the FSH secretion. So faster for LH and slower frequencies for FSH. Continuous administration of GnRH abolish both LH and FSH secretion. Therefore, superactive GnRH analogs like goserelin, triptorelin, leuprolin are used to suppress the gonadal function. So GnRH analogs basically are goserelin, triptorelin, and leuprolin. In the pituitary, LH um, from the pituitary, LH secreted, which drives the interstitial cell synthesis and secretion of testosterone in both sexes and triggers ovulation in female. Negative feedback by the sex steroids. So LH is basically inhibited by the sex steroids. FSH in male drives Sertoli cell-derived normal sperm maturation and production of seminiferous tubule fluid, as a number of substances thought to be important for spermatogenesis and inhibin. FSH in female drives granulosa-mediated aromatization of the androgens to estradiol. Negative feedback is mainly by the inhibin. So basically, LH is important for the testosterone production in both sexes. In female, LH triggers ovulation. It is inhibited by the testosterone or other sex hormones, sex steroids. FSH basically helps in the sperm maturation and ovulation, and it is basically inhibited by the inhibin. FSH in female is important for the production of estradiol. Like in male, it is the LH. However, in female, basically FSH helps in the aromatization of androgens to estradiol, and it is inhibited by the inhibin. So gonads, in the gonads, testosterone and androstenedione and dihydrotestosterone are produced by the Leydig cell. So three hormones are produced by the Leydig cells, including the testosterone, androstenedione, and DHT. Male testosterone has a circadian rhythm. The maximum secretion occurs at 8 a.m. and minimum at 9 p.m. Female testosterone production peaks at ovulation. Estradiol production in male, mainly from the peripheral conversion from androgens. In female, estradiol rises with follicle maturation under the FSH drive of the granulosa cell. Estradiol is made by the placenta and corpus luteum. Progesterone is secreted by the corpus luteum, which is derived from the granulosa cell only after ovulation occurs. Peak production is on 21 day of the 28-day cycle. So regulation of gametogenesis, both FSH and LH are required for the gametogenesis. LH ensures high intragonadal concentration of testosterone. Exogenous testosterone cannot be substituted. FSH, though its action on the Sertoli and granulosa cells is vital for the sperm and oocyte maturation. The whole process of spermatogenesis takes around 74 days. It's followed by another 12 to 21 days for sperm transport through the epididymis. This means that the events may as a whole take for up to 3 months. In the successful induction of spermatogenesis, treatment may take up to 2 years. In female, from primordial follicle to primary follicle, it takes 180 days. That's a continuous process. It then takes another 60 days to form a pre-antral follicle, which then proceeds to ovulation 3 menstrual cycles later. Only the last 2 to 3 weeks of this process is under the gonadotrophin drive, during which time the follicle grows from 2 to 20 millimeter. So the reproductive physiology, testosterone is daily produced at a rate of 5 to 15 mg per day in male. 2 to 4% of the total testosterone is free. The rest is bound to sex hormone binding globulin, and androstenedione is only 6% is sex hormone binding globulin bound. Estradiol production in female is around 40 to 400 micrograms. 2 to 3% is free and is biologically active. The rest is bound to sex hormone binding globulin. So in male, the testosterone is basically 5 to 15 mg, 2 to 3% of the total testosterone is free. While in case of female, the estradiol production is 40 to 400 microgram, much less than the testosterone, and 2 to 3% is free. So sex steroid metabolism, testosterone is converted in the target tissues to more potent GHT in the presence of 5-alpha reductase. There are multiple variants of the 5-alpha reductase isozymes. Type 2 is the isozyme responsible for DHT synthesis in the genitalia, genital skin, and hair follicles. So type 2 DHT is basically involved in the development of the secondary sexual characters. It is therefore for the normal important for the normal male